sphincter is divided into 3 compartments. Duodenum is about 20 centimeter, jejunum 2.4 meter, and the last one is the ileum 3.4 meter. Surface area of the small intestine greatly increased by the intestinal mucosal foldings, villi, and microvilli, as you know from your previous studies in the anatomy. Also, site of greatest amount of digestion and absorption of nutrients and water. All these events take place in the small intestine. Okay. Here we will deal with the modifications to increase surface area. There is 3 structures in the small intestine increase the surface area. Increasing the surface area increase the efficiency of the absorption, because as you know, the main function of the GIT in general is the digestion and absorption. This is why when you increase the surface area, that means you increase the efficiency of absorption. There is 3 compartments. Number 1, biliaris circularis, is a circular folds, as you see in the picture. Number 2, we have what is called villi, that contain capillaries and lacteals, folds for mucosa. And the 3 one, or the last one, 3 is microvilli, that is folds of the cell membranes of the absorptive cells. These 3 compartments increase the surface area, increase the efficiency of absorption. Slide 5, secretion of small intestine. Secretion of the small intestine composed main 3 substances. Number 1, alkaline fluid, primarily composed of water, electrolytes, and mucus. The volume of fluid is about 1 to 1.5 liter per day. Mucus, the importance of mucus is to protect against digestive enzymes and stomach acids. Number 3, digestive enzymes. They are proteins responsible for the digestion of many foodstuffs. They are bound to the membranes of the absorptive cells. For example, disaccharides or disaccharidases, peptidases, and nucleases. In addition to others, for example, maltases and something like that. All these enzymes are included in the intestinal secretion, along with the mucus and alkaline fluid. Here we have secretion. As you know, the duodenal secretion, or the duodenum, is lined by the Brunner's gland, which is responsible for the release or for the secretion of mucus. Mucus rich or mucus containing bicarbonate. This bicarbonate is so important to neutralize the acidity which is come from the stomach, because the first part of the small intestine is rushed by the acidity from the stomach. As you know, the first part is the duodenum. This is why the Brunner's glands here is so important to prevent the epithelial cell of the stomach from the ulceration. Also, we have Pani's gland. The Pani's cells are found in the lumen of these crypts. According to today's knowledge, they play a role in the antimicrobial defense. Why? Because the Pani's cells secrete what is called enteric alpha defensin. Okay, From this above mentioned, you can know the function of the Brunner's glands. It have a role in the protecting the duodenum from the damaging action of the acid, as Brunner's gland area is more resistant to acid than lower regions of the duodenum. Okay, now we will see the composition and properties of the secretion. The choice is clear or yellowish and is rather viscous as a result of the presence of mucoprotein. This mucoprotein is make it viscous. The sodium and potassium concentration are also close to those of plasma. The anion are chloride 136 millimole per liter, and the bicarbonate 14 to 40 millimole per liter, and are independent of flow rate. Here is the regulation of secretion. How the GI regulates or how the small intestine regulates the secretion of it. Spontaneous secretion occurs and the act of the feeding initiates or increases the rate of secretion. قالوا البيزل secretion اللي هو السبونتينيوس الاماونت بتاعته قليله جدا، لو لاحظتها approximately 1 to 2 ml per hour, little amount of secretion. But when you ingest something, increases to from 5 to 8 millimole or ml per hour. This high increment due to ingestion or due to feeding. That means feeding can increase the secretion of a small intestine. Both neural and humoral mechanisms are involved in feeding stimulation. Feeding stimulation can occur through the neurons or through the humoral. Humoral is hormones. Regulation of secretion, also we are dealing with the regulation. The most important factor that regulates secretions in the small intestine is the presence of chyme. The chyme is the semi-solid content of food that enter the GIT. This is largely local reflex. Why is local reflex? Because the small intestine is surrounded by the smooth muscle. As you know, one property of the smooth muscle is the stretchability and contractility. This is largely local reflex action in response to chemical and mechanical irritation from the chyme and in response to distension of the intestinal wall. When you distend, the smooth muscle is retained back to previous condition. Also, regulation of intestinal secretion can be controlled by vagal stimulation. The vagus nerve in the GI, as you know, and then from your neuroanatomy knowledge, the vagus nerve is mixed nerve, sensory and motor. Vagal stimulation increases secretion rate, but some scientists say that crude secretin is a hormone. Preparation stimulates secretion, but not pure secretin. Similarly, crude gastrin preparations were effective stimulants, but pure gastrin was not. As we said before, in addition to Brunner's glands in the small intestine, especially in the duodenum, we have also intestinal epithelial cells. The surface epithelial cells of the gastrointestinal tract also play a protective role by stimulating bicarbonate secretion. As you know, bicarbonate is alkaline, and the HCl from the stomach is acid. When it reacts together, it leads to neutralizing the acidity, because the harmful effect of acid can lead to ulceration, especially if the if there is something problem in the mucus secretion. This is why the bicarbonate secretion is also so important. Using pH sensitive microelectrode, a pH gradient has been demonstrated so that with the lumen at pH 2, the pH in the vicinity of the mucosal cell membrane was 7. This little bit is a huge difference, because in the presence of the bicarbonate, the pH is increased to reach 7 from 2. That means the difference is 5. pH is not easy. All this difference due to presence of bicarbonate in the intestinal lumen. This gradient is set up by the secretion of bicarbonate into the surface gel. Here we are dealing with secreting eggs. When we say secreting eggs, we mean by that anything that stimulates the secretion is called secreting eggs. Anything lead to increased secretion is called secreting egg. Okay? Stimulation of bicarbonate is brought about by a number of hormones, including gastric inhibitory peptide, GIT, stimulate bicarbonate, pancreatic glucagon, the antagonist of insulin, as you know, also stimulate bicarbonate secretion. Adrenaline and noradrenaline, stress hormone, also responsible for the bicarbonate secretion. Luminal pH is an important factor in the regulation of bicarbonate secretion also. That means when the pH is increased, the secretion of bicarbonate is decreased, and vice versa, which operates in the mammal with prostaglandin as one of the mediators. This process of this glucagon of these hormones is mediated by prostaglandins. Indeed, arachidonic acid and a number of prostaglandins. Arachidonic is the precursor of prostaglandin, as you know from your biochemistry background. Prostaglandins stimulate bicarbonate secretion, with E series of prostaglandins being the most effective, because we, as you know, we have a different kind of prostaglandin. The most important one in the bicarbonate secretion in the intestinal secretion is the prostaglandin E. Is the most effective. Inhibitors of prostaglandin synthesis reduce bicarbonate secretion. Dibutyryl cyclic adenosine monophosphate and phosphodiesterase inhibitors also stimulate secretion, and it has been suggested that the prostaglandins might operate through cyclic AMP, one of the most famous second messenger, as you know. Slide 15. Experiments reveal that exposure of the mucosa to the toxin for longer periods stimulate the adenylate cyclase of the crypt cells and those of the villi. As you know, the adenylate cyclase stimulates the cyclic AMP, because it's responsible for the conversion of ATP to cyclic AMP. These experiments suggest that the cyclic adenosine monophosphate formed from the hydrolysis of ATP, adenosine triphosphate, by adenylate cyclase enzyme in the cell membrane has a dual function. Number one, in the villus cell, it prevents absorption, whereas in the crypt cell, it induces secretion. This is why they say it with a dual action or a dual function. Parasympathetic activity increases intestinal secretion, whereas inhibition occurs with sympathetic stimulation. As you know, in many instances, the parasympathetic is the antagonist of sympathetic. The direct application of acetylcholine to sheets of intestinal mucosa in vitro stimulates electrolyte transport as an index of secretion. These effects are blocked by atropine. As you know, atropine is the antagonist of acetylcholine, which is released by the parasympathetic. And also it's considered as a parasympathetic neurotransmitter. These effects are blocked by atropine, and therefore muscarinic receptor is involved. Alpha-adrenergic receptor or alpha-adrenergic agonist, on the other hand, enhance absorption and inhibit secretion. As we said before, in most cases or in most conditions, parasympathetic is antagonist for the sympathetic. VIB hormone or VIB is the vasoactive intestinal peptide. Also is a hormone that stimulates intestinal secretion in both small intestine and large intestine. Perhaps the most spectacular intestinal secretagogue is a cholera toxin. As you know, is one of the bacterial toxin which stimulates the secretion through the cyclic A and B mechanism. You can see the chapter 1 in physiology to understand the cyclic A and B, or to remind how the second messenger cyclic A and B is working. However, recent evidence has been presented that the toxin may have its effect, at least in part, through the enteric nervous system. That means the cholera toxin affects the cells of the mucosal cell and also affects the enteric nervous system. Bile acids and fatty acids. Both of these agents have been implicated in intestinal secretion and diarrhea. The bile acids are particularly effective when deconjugated. As you know, we have conjugated bile acids and deconjugated bile acids. The deconjugated is more effective. Their effects are mediated at least in part through the increased cellular cyclic A and B. Also, the second messenger here in bile acids is cyclic A and B. Here we can summarize the agents known to stimulate intestinal secretion. We have two columns. The left column is titled as agents acting from the lumen. The right column titled agents acting from the serosa.
والله بلد الفوضى. أتيجي أوريك هالمصير الحلويات
في كلام
بالرجال رخيصة في مصر. ورقة الصبح حيث يجي فيه بدأت راعي بالحلويات. جابها من مصر. مصر ده.
السلام عليكم.
يوم المسارة سودا درما.
شباب.
كويس علبة.
أنا كنترول على السبيكينج. شكرا لك. طيب. هي قلنا عندنا summarization للاجنتس acting from the lumen في الليفت سايد او في الليفت كولم، والرايت كولم عندنا agents acting from the serosa. Serosa, that means these agents go through the bloodstream, while lumen, as you know, in the intestinal cavity. Bacterial toxin secretion or secretion, bile salts, and fatty acids. All of them are increased secretion through the lumen. From the through the serosa, that means it's going through the bloodstream. It's vasoactive intestinal peptide, it's the hormone, prostaglandins, acetylcholine, 5-hydroxytryptamine, substance B, neurotensin, calcitonin, vasopressin. All of them are hormones. This is why their pathway is through the blood. Here we have a region about the second messenger to understand how the secretory function of the small intestine is controlled by two major second messenger systems, cyclic A and B and calcium. As you know, the calcium can act as the second messenger. Vasoactive intestinal peptide and cholera toxin induce secretion by and to revise how the cyclic AME is facilitate or induce the action of many substances as a first messenger. As you see here, in the extracellular fluid, we have a first messenger. This is the ligand protein, transmembrane protein, protein relay. This is protein. This is adenyl cyclase enzyme, right, as effector, change the ATP to the cyclic A and B, and the cyclic A and B give a physiologic action. Sometimes it's a metabolic, and sometimes it's a structural change because it's induced protein synthesis inside the cell. Here, this slide shows the second messenger of calcium. As you know, from the diacylglycerol, the plasma membrane, in presence of IP3, inositol triphosphate, inositol triphosphate bind its receptor in the endoplasmic reticulum. As you know, endoplasmic reticulum considered as the main source of intracellular calcium. This binding of IP3 with its receptor release calcium from endoplasmic reticulum, and the calcium from the endoplasmic reticulum in the cytoplasm go directly to the Golgi apparatus and assimilate diacylglycerol through the protein kinase D and protein kinase C, which give us the physiologic action. Acetylcholine and 5-hydroxyamine are natural secretagogues and utilize the other major pathway based on intracellular calcium, whereas the intestine has receptor for acetylcholine, which are analogs to the calcium mobilizing receptors found in the other cells. The calcium mobilizing receptors are responsible for a number of possible second messengers, such as calcium cyclic guanosine monophosphate and arachidonic acid metabolites, for example, prostaglandin, thromboxans, prostacyclines. From here you can see and you can note the importance of the second messenger. In summary, three parts of the small intestine are the duodenum, jejunum, and ileum. Intestinal secretion contains water, enzymes, electrolytes, and mucus. Mechanism of secretion regulated by many factors such as feeding, humoral, and neural agents. The secretory function of the small intestine is controlled by two major second messenger two major second messenger systems, cyclic A and B, and calcium. By the end of this slide, we are finished. This is the reference. If you have any question, any comment, any addition, you are welcome. في سؤال، في استفسار، أو في شيء ما واضح، أو في إضافة؟ ما في سؤال يا شباب؟ أو ما في واحد داير يضيف لنا حاجة؟ طيب لو ما في سؤال نكون خلصنا محاضرتنا إن شاء الله. نتمنى إنه الجميع استفاد، وربنا يعينكم إن شاء الله. في أمان الله وحفظه. السلام عليكم ورحمة الله.
عليكم السلام ورحمة الله.